Modern neurobiology maps depression as a physical dysfunction of discrete neural circuits, localized primarily within the medial prefrontal cortex. Standard monoamine therapies operate on systemic levels, attempting to shift brain chemistry broadly. These drugs require weeks to reorganize network connectivity, a delay that often leaves patients without relief for a month or more. Ketamine proved that circuit repair can happen on a much faster timeline. A single dose can restore coordinated activity in the prefrontal cortex and reverse depressive phenotypes in less than 4 hours. However, this efficacy is currently tethered to severe clinical limitations. Ketamine induces intense dissociation and hemodynamic instability, and its interaction with the brain's reward systems creates a high potential for abuse. Ketamine is best understood as a pharmacological probe rather than a final treatment. By tracing exactly how it repairs these circuits, we can identify the specific cellular sites required to design a new generation of targeted therapeutics. To engineer a better alternative, we have to resolve a core pharmacological contradiction. Ketamine is an NMDA receptor pore blocker. Since NMDA receptors are excitatory, blocking them should theoretically diminish neural activity. Yet following administration, we see a paradoxical surge in the firing rate of excitatory pyramidal neurons in the prefrontal cortex. This indicates that ketamine acts through disinhibition. It must first silence the local inhibitory networks that normally suppress the primary excitatory neurons, effectively releasing the brakes on the circuit. Somatostatin expressing, or SST+, interneurons are the specific gatekeepers in this region. They are the regulatory cells that ketamine must bypass to initiate its effects. Isolating the molecular mechanism that inhibits these specific interneurons provides the structural blueprint for creating rapid-acting circuit repair without ketamine side effects. In the microcircuit of the prefrontal cortex, SST+ interneurons project their axons directly onto the apical dendrites of pyramidal neurons. This physical relationship allows them to dictate synaptic integration, exerting targeted inhibitory control over the pyramidal neuron's electrical input. Chronic, unpredictable stress breaks this regulatory balance. Sustained stress physically alters the architecture of these interneuron connections. Stress drives presynaptic hypertrophy. The SST+ axons thicken, and the density of their synaptic boutons multiplies aggressively along the pyramidal dendrite. This expansion creates an unyielding inhibitory clamp. The pyramidal neuron is forced into a state of chronic suppression, which prevents coordinated ensemble activity across the prefrontal cortex. This sustained inhibition leads to the starvation and retraction of dendritic spines on the pyramidal cell, as it loses the active synaptic connections needed to maintain its structure. This micro-level atrophy correlates directly with observable depressive behavior in both rodents and humans. To reverse these symptoms, a therapeutic must physically break this hypertrophic clamp, allowing the pyramidal network the freedom to rebuild its structural integrity. Ketamine achieves this by targeting the overactive presynaptic boutons of the SST+ interneuron. The process is initiated by μ-opioid receptors, or MORs. While ketamine's primary target is the NMDA receptor, its rapid antidepressant effects rely on MOR activation. These receptors are specifically enriched on the presynaptic terminals of the SST+ interneurons, precisely where the inhibitory clamp originates. Ketamine acts as a partial agonist at these sites, triggering an intracellular GIGO signaling cascade within the SST+ terminal. As the signaling cascade activates, neurotransmitter release is rapidly suppressed. The hyperactive interneuron enters a transiently inhibited state, visible in this sharp downward spike in activity. Silencing the interneuron immediately lifts the inhibitory clamp, allowing the pyramidal neuron to fire at a normal, coordinated rate. This surge in pyramidal activity triggers the release of brain-derived neurotrophic factor, which activates the mTOR pathway. This molecular bridge drives the rapid regrowth of the dendritic spines that were pruned away during chronic stress. This mechanism is highly effective, but because MORs are expressed throughout the brain, targeting them systemically is what creates ketamine's significant abuse liability. The engineering goal is to inhibit these specific interneurons without engaging the opioid system at all. This requires shifting from structural anatomy to molecular mapping, identifying other receptors that are uniquely expressed on SST+ interneurons. Researchers used cell-type-specific RNA sequencing to isolate the molecular profile of these interneurons and compare them to the surrounding pyramidal population. They focused on G-protein-coupled receptors, or GPCRs, the molecular actuators that dictate how a cell responds to its environment. The sequencing revealed an ensemble of GPCRs that are highly enriched in SST+ interneurons, creating a targeted molecular fingerprint for this specific cell type. This transcriptomic map provides a list of alternative targets that could theoretically achieve the same circuit disinhibition as ketamine. At first, the solution seems simple. Pick the most enriched receptor on the map and hit it with a high-dose drug. But neuromodulatory receptors are rarely exclusive to just one brain region. To reach a therapeutic dose in the prefrontal cortex, a single drug often requires a high systemic concentration. This high concentration triggers receptor activity in off-target regions, leading to the same types of physiological and behavioral side effects seen with ketamine. Relying on a single receptor often just replaces one set of side effects with another. To solve the localization problem, we have to move towards synergistic polypharmacology. We must find a way to achieve massive circuit-level inhibition without ever using a single high-dose compound. This is the logic behind the SST plus cocktail, a treatment that simultaneously targets 3 different receptors co-expressed on our target interneurons. Using the RNA-seq data, researchers combined an mGlu-R1 modulator, a 5-HT1A agonist, and a partial MOR agonist. Crucially, each individual drug in the cocktail is administered at a strictly sub-threshold dose. Injected on their own, these low doses produce no therapeutic effect and remain well below the level needed to cause side effects. But because the SST+ interneurons are the only cells that co-express all 3 receptors at high levels, the cocktail converges on this single cell type. The simultaneous activation of these 3 receptors triggers an additive GIO signaling event. The signal crosses the threshold exclusively within the target interneuron. Synergistic targeting multiplies the drug's efficacy at the exact site of the circuit pathology without increasing the drug concentration in the rest of the body. The behavioral data confirms it. When combined, this sub-threshold cocktail rapidly reverses depressive behaviors, matching the efficacy of ketamine. Most importantly, the side effect threshold is never breached. Because the receptors aren't co-expressed elsewhere, the low doses fail to trigger off-target effects. By successfully inhibiting the interneurons, the cocktail permanently lifts the inhibitory clamp on the pyramidal network. The resulting surge in neural circuit activity rapidly regrows the dendritic spines that were lost during chronic stress. We have successfully engineered the circuit-level cure of ketamine without the dissociative symptoms or the risk of abuse. This strategy of mapping cell-type-specific transcriptomes to drive synergistic polypharmacology provides a new blueprint for the rational design of safe, rapid-acting psychiatric medicine.